So in the Miller lab, we're interested in shortening the diagnostic odyssey for patients with genetic disorders. This workup is systematic and can take years to complete. And at the end, only about half of individuals end up receiving a precise genetic diagnosis. We believe that new technologies such as long-read sequencing can both shorten the diagnostic odyssey and increase the rate of genetic diagnoses. This lowers barriers to accessing comprehensive genetic testing. And results in better outcomes for patients and their families, and it may uncover novel mechanisms of disease that we can study in the lab. So today our efforts are focused on using Nanopore sequencing to analyze challenging unsolved cases and develop new clinical tests. Nanopore sequencing works by detecting changes in current as a single-stranded DNA or RNA molecule passes through a protein pore. These current changes are characteristic, so we can work backwards to determine exactly what nucleotides are in the pore at a given time. Importantly, methylated nucleotides have a different current profile than unmethylated ones, so we get not only sequence data, but modification as well. I'm sure this is all quite familiar. I'm going to skip over most of this case study for time, but the point of it is just to say that the advantages of long-read sequencing are that we can use SNPs, indels, structural variants, and methylation all from the same source, and all of those things together help us to solve cases that otherwise we can't. And methylation is a really important part of the puzzle in cases such as this one. If you'd like to discuss this specific case, you can talk to me afterwards. So methylation regulates gene expression, and the patterns are typically inherited a lot of the times differentially paternally than maternally, and they are tissue-specific patterns. CPGs are distributed all throughout the genome, but because of that, that makes interpreting the raw data right when you get it for the whole genome rather difficult. You can look at a region like this, and it looks kind of like a mess. So what I found is we need to break it down into smaller regions, at least clinically, that we already have established meaning for. So CPG islands are stretches of DNA with a CG to GC ratio greater than 0.6, and we often see those near promoter regions. The need to quickly identify these differentially methylated regions throughout the whole genome drove me to develop the methylation optimization wizard, or MeOW. The goals are pretty simple. The input should be an aligned BAM file containing methylation tags. Those tags, the output of it should simply be differential methylation at each of those, within each of those predefined intervals. It should be pretty fast, and ideally at the end of it, you should end up with nice figures that can go directly into presentations. The way that it works is you are testing alignments within a data set against one another. All of the samples really do need to be from that same tissue source for this program to work well. All of the ones in this example are from blood. I suppose you could use it to test tissues against one another if you wanted. The sequencing depth also needs to be fairly high, so around 30x coverage is what I use as my cutoff. It's most important that it's consistent across all of the samples that you test. The input BAM files need to have CPG methylation encoded using HTS5 standards. I do try to keep it updated. And then the first part of it is a command line utility. That utility generates a database of CPG methylation frequency within defined intervals that you supply. So those for me were CPG islands defined with that 0.6 ratio, but you could use whatever you want and make your own intervals. And then those intervals should be annotated with the gene names to make your analysis easier. And then I would obviously recommend assigning an identifier. Once you have that database, we move over to R, where I've made my library. So in R, any one sample in the database can be tested against all of the others across all of the intervals all at one time. Test statistics are corrected for the enormous amount of multiple comparisons that we're making, and I use Benjamin Hochberg to do that by default, but you can use whatever you want within R. And I do offer 3 different test statistics options and additionally calculate effect size, which will help you filter your enormous amount of results. So the reason that I have 3 test statistics is because the distribution of methylation, even within these CPG islands, is not very homogeneous over the space. So MeOW can calculate a pooled variance test, and the effect of that would just be to ignore that wave pattern that is so obvious to us, and just look what is the difference between the gray band, which represents all of the reference sample, and all of the red cloud of dots, which is the test sample. Or you can do a paired test, and you can assume that maybe the shape of that wave is important, and we want to see the difference in the shape moved up. The third version we'll talk about later. All the tests are bootstrapped, which ensures that each comparison is including variability in the full control data set, but that we're also using the same number of positions across all CPG islands. So it should be a fair comparison between all of them. The extra special test statistic is fitting it onto a beta distribution. And the reason that I did this is the beta distribution is a better fit for the 0 to 1 interval than a t-test is, which assumes a normal distribution. These means comparisons are in that bounded interval, and I'm using moment estimation for it. I have another version, which is optional, takes much longer, where you actually fit a distribution to within each CPG island, but it takes a very long time. And then the effect size for everything is Cohen's d, and that's used to filter out extremely consistent but extremely meaningless differences. All right, so once you run all of this, we have a function that produces a database that you then filter and analyze. For most of my samples where there actually is something, I'm putting in like Prader Willi, a sample that I know has an imprinting disease, it will go across all 28,000 CPG islands that meet my my size criteria down to between 2 and 10 candidate differentially methylated regions. And you make nice looking charts. So here's an example with 2 well-known imprinting disorders, Prader Willi and Angelman syndrome. So in this collection of syndromes, a small region of chromosome 15 is differentially methylated depending on the parent of origin. So both syndromes have multiple causes. You can have, it can be caused by SNP, uniparental disomy. There's a broad array. So to look at this one, I took DNA from the blood of 20 individuals with either Prader-Willi syndrome and Angelman, and compared it against 20 controls, people know not to have this, just to see if my tool could pick it up. We'd find the right differentially methylated CPG. So in this example, the test was sequenced to a depth of 32x. This is all in the Promethean. Our read lengths are average 30 to 40 for this study. The CPG islands to go through the test have to have been sequenced in both the reference and the test sample. For this one, I required everything that I tested to have at least 100 positions. I often use this with 50 positions, and it works fine. So I filtered my results with an effect magnitude of 1.5, my BH-corrected p-value to 0.01, and then also the mean difference of 30% going upwards, because this is clinically relevant. And I ended up with only one candidate. So it correctly picked out SNRF-SNRPN, which is the gene that is known to be associated with Prader-Willi. You can confirm this by visualizing some of the other Nanopore data sources. So on top, I have the coverage plotted, and we can see that we have a very, very large deletion. And then on the bottom half, I have CPG islands plotted. The red is the sample, and the gray is the reference. And you can see that we have increased methylation in several CPG islands within these genes, but you can probably pick out the one which is the significant one. We've also tried this with a few unknown cases where we didn't know whether they had, which one they had, Angelman, Prader-Willi. And it does pretty well on that also. This one did identify some other genes, which I don't particularly know the meaning of, and we haven't really found any great associations there, but it also found the right one. If we look at those in the other format, so not in the box plot form, then I think that you can convince yourself that that's definitely a false positive, and that these, even in the control data set, it's not a very definite area. So it will always require some level of human vetting. It won't get you all the way there, but it does get you much closer. And then on that same sample, when we looked, you can confirm again that there's a loss of methylation instead of gaining methylation here. So this is a case of Angelman's. So in the future with this tool, I'm hoping to incorporate phasing and haplotype-specific methylation so that we can expand this to other cases. I'd like to validate it with other tissue types that we see often in our lab, like fibroblasts and buccal swabs in particular, which I think will be challenging. And then I'm very excited about exploring other uses for this, which I think could be pretty big within cancer research in particular. And then, of course, I'd like to distribute R's library so that other people can use it. I'd like to acknowledge everyone in my lab who are very supportive and have helped me a lot in developing all of my research.